Good morning. This is Boyd Biology, meiosis and Mendel. We've talked about mitosis, we've talked about meiosis. Now let's talk about Mendel. And it's going to be kind of like going back in time a little bit. We entered well into the first half of the 20th century regarding our biological understanding. Cell biology is very much underway. The discovery of DNA as the inheritance molecule, all the experiments that led up to that. But to really get a good handle on inheritance, which of course we know now that genes are passed on through gametes, haploid gametes produced by reproductive cells. Sperm and eggs are the gametes. And you're going to have one of each chromosome from each parent. The sperm cell has 23 chromosomes. The egg cell has 23 chromosomes. And you're going to get a set of each. You're going to get a pair total, one from each parent. And so how do those pairs interact with each other? What really is a gene? How can you have different variations of a gene, like eye color or attached or unattached earlobes, or the ability to curl your tongue or not? These are all inherited traits that are coded for in your genes. But there's some other important patterns to understand regarding how inheritance works. And so inheritance has been something that people have studied since the beginning. We notice patterns. We notice when offspring are like or unlike their parents. This is obvious just in any kind of walking around town or observing of nature in a casual way, but it's incredibly obvious if you're involved in breeding of animals or plants. So if you're in agriculture, if you have a garden, if you raise any animals at all, and most humans over the history of the world have been involved in that process, these patterns become very apparent. And so Gregor Mendel, who is kind of the hero of our chapter. This is the finishing or the completion of the chapter in Novare, chapter 6, and it's the new chapter 14 in Riot. And so we're going to be talking about Mendel. We're going to get some background on Mendel, who took these patterns that he saw and started to actually apply mathematical probability. If I bred these two organisms, what are the chance I will get these traits in the offspring? And are there any observable, quantifiable patterns? Mendel really broke the mold, and so we're going to talk about how he did that, and really making this connection between meiosis and Mendel, remembering that we're in reverse order chronologically. Mendel's work was late 1800s. And he didn't have a good sense for chromosomes or DNA. None of this stuff had really been discovered. Cell biology was well underway, but regarding how this worked at the cellular level, he didn't have an understanding of it. So his observations and his science were at the organismal level. He was studying what he could see in front of him. He was studying traits he could observe in organisms. And so it's kind of an interesting confirmation of what we know about meiosis beforehand at the organismal level from this remarkable person, this friar named Gregor Mendel. And so let's head to the slides. Okay. So Gregor Mendel, an Augustinian friar. I've talked about this with a couple of folks. Most textbooks call him a monk. I remember about a decade ago, the Field Museum in Chicago put on a wonderful Gregor Mendel exhibit. And I was in touch with the person who produced that, and he forwarded along some helpful resources for teaching. And he insisted that evidence shows that Mendel was not truly a monk. He was a friar, and that's kind of a different type of position within the monastery, more of a teaching fellow of sorts. And so this is the kind of the Austria-Hungarian Empire at the time, and this is late 1800s when Mendel was there. and doing his work. And Mendel was an interesting character. He really had an interesting upbringing, spent a lot of time studying plants and raising fruit from fruit trees with his father, grafting the trees and doing all kinds of interesting orchardist types of work. And this that foundation of interaction with the landscape, interaction with creatures in real life, getting his hands dirty, understanding and studying and watching plants, gave him some of the most important foundation that led to his scientific contribution. And so he eventually went on to publish his work, which was studying pea plants. 8 years of studying, planting, collecting data, observing pea plants, and then recognizing and discussing and quantifying the patterns of inheritance regarding certain traits that were passed on by these pea plants. So 10,000 pea plants, that's a lot. And he really connected the dots in a remarkable way. Up until this time, most scientists, most biologists, I should say, really kind of kept math at a bit of an arm's length. Math's great, but it's not too helpful in this work we're doing. This is natural history or natural philosophy. We're just describing creatures and their life cycles and observing different things about them. We don't really there wasn't seen a need really necessarily for math, or no one had connected the dots as to how math could be useful in describing patterns in biology. Mendel saw this up close and personal. He had scientific training at university. He had a strong mathematics background. And so when he started to conduct his pea plant experiment, he most certainly saw where math fit into describing unique biological phenomena, and especially describing patterns over time. Probability and statistics now are absolutely fundamental for anyone going into biology. Almost any single published paper makes use of some type of statistical analysis, which attempts to explain the findings. And in Mendel's case, attempted to explain his findings regarding the probability of certain traits being inherited from ancestors, or from the parent generation to the what's called the F1 generation, the P1 generation of the parents, and then the first filial generation, F1, and then the second, F2. And so what did Mendel do? And maybe I'll back up just a second before we start talking about peas. Let's just talk about bears. Let's talk about polar bears and grizzly bears. We know that polar bears and grizzly bears can hybridize, and maybe we'll go back to this screen so you're not thinking too much about what's happening there with the peas. Polar bears and grizzly bears can interbreed and produce fertile offspring. Unfortunately, they call them a pizzly, which is a little bit silly, but hey, that's okay. It's kind of a silly idea. These organisms typically don't interbreed, but where their ranges overlap, there have been documented examples. There are zoo examples as well, and those offspring are often fertile. Okay, and so we would think of a brown bear or a grizzly bear being a true breeding brown bear, right? All the genes that it's going to pass on in its sperm or eggs are going to be brown bear or quote unquote grizzly bear genes. Ursus arctos horribilis. Okay, and the polar bear is going to pass on polar bear genes, right? You've got a polar bear and a brown bear. They're quote unquote true breeding. And Mendel used that word true breeding to describe his peas, but it's a little bit easier to see first in something as big and obvious as a bear. So we have a brown bear father and we have a polar bear mother. Okay, and the brown bear is going to pass on brown bear genes through those gametes, one of each chromosome in each sperm cell of that brown bear, and the polar bear female is going to pass on one chromosome of each pair in that egg cell, and the offspring are going to inherit the set. The offspring are going to get half of their chromosomes and genes from mom and half from dad, and those chromosomes interact with each other. You have homologous pairs, right? You have chromosome 1, you have chromosome 13. Both of those pairs of chromosomes are going to have codes for the same gene on them. But why do we have variation then? Why do we see variation in traits? Why do we have blue eyes and brown eyes? Why can a parent have brown and a child have blue? Why can a parent roll their tongue and the child cannot? That's because there's a little more to the story than just the gene itself. There's variation of form within a gene. And so that's a key concept in this first lesson. And that's why we're bridging the gap between meiosis and Mendel. Is I want you to connect the dots here. What we see in these little variations, and I'll go ahead and pull up that slide and we'll come back to peas. I want to continue this example with bears for now. If we look at the architecture or structure of the chromosome, here it is. Like the language of inheritance. Remember, Mendel doesn't know any of this stuff. So this is fast forwarding back to where we were in the mid 20th century. Here's a chromosome pair. And let's consider the grizzly bear and the polar bear first. Okay, and this says for flowers, but let's just pretend it's talking about fur color. All right, those offspring, let's say the polar bear female, the grizzly bear male, they mate, they have twin cubs. Twin cubs are born, and they each received a set of chromosomes from each parent. And so we're looking at, let's say, chromosome 4 of this Pisley offspring. Okay, they're each going to have 2 of each chromosome pair, one from mom, one from dad. And let's say this one's from dad, and there's the gene for fur color. Dad's chromosome, the gene for fur color. This is the locus or location of that gene. And we've sequenced a lot of these things. We know with very, very high degree of precision where certain genes are on which chromosomes, such as the hair color on the bear chromosomes. Okay, so here's dad's brown fur gene and mom's white fur gene. Same gene, very same location, different expression of that gene, right? So polar bears and brown bears can interbreed and produce fertile offspring. That means they're very similar genetically. So it's the same gene, but it's a different version, slightly different code, slightly different expression. It's still color of fur, but we get white, quote unquote, white fur from mom. We get brown fur from dad. This is a hypothetical example. And so those are called alleles. Dad has the allele or the code for that gene for brown fur. Mom has the code for that gene for white fur. Those two will interact, and a couple of possibilities arise. And it could be that the offspring have a mixed fur color. It could be that the offspring have a brown fur or a white fur. Those are the possibilities. Okay, now let's take this back and consider pea plants. Why did Mendel pick peas? Peas are going to be easier to study than populations of brown and polar bears. They're easy to cultivate. They grow quickly. They're hardy. They can grow early in the spring, but you can grow multiple generations of peas each growing season. And so they're easy to cultivate. They aren't very picky about the soil or the weather, and they're easily observable. And they're contained in one space. Perfect model organism for understanding inheritance. And you guys have read a little bit about model organisms. Model organism, of course, being an organism that's easy to study, to observe certain patterns. And this is no exception here, the pea plant. And so Mendel noticed some differences about his pea plants. He planted his first crop, let's say, and this is just hypothetical. He planted his first crop and he started to notice certain things about them. Some of them are tall and some of them are short, and there doesn't seem to be an in-between. And so these he called definable characteristics. And so he started to play with breeding of these a little bit. When he bred a tall with a short, he almost always got all tall offspring. That's interesting interesting situation. These plants are breeding true. They're breeding true. The tall plants always give us tall offspring in this case. These true breeding plants. He knew that this tall plant came from tall parents. He knew that this short pea plant came from short parents. When he bred them together, it was always tall pea plant offspring. That's interesting. So there seems to be an interplay between these characteristics. One trait, and I'm going to introduce a lot of vocabulary today. One trait seems to be dominant to another. Tall seems to be dominant to short. In other words, whenever we breed these true breeding plants, they always are tall, even though one parent is short. Well, that's interesting. And so we would call tall dominant. We would call short recessive. And so dominant traits mask the traits of those recessive parents. And so Mendel didn't use these terms yet. He's still observing patterns. So I'm putting terms into use that he developed later and that other people developed later. But he noticed this phenomenon. There seem to be pairs of traits, one or the other, no in-betweens. And when you bred certain individuals with certain individuals, you got all of one. And that seemed to be true for all of these traits that he looked at. That was true for height, we see here. Tall seemed to be dominant to short. It seemed to be true for where the flowers grew on the plant, whether the flowers were terminal or whether the flowers were axial in the axes of the branches. One of those was dominant over the other. For color of the pod, for shape of the pod, for color of the flower, color of the seeds, and for shape of the seeds. And so all of these traits, true breeding traits that seemed to always have one or the other, no intermediate forms. So it occurred to him, we get equal information. He didn't know the terms chromosomes or DNA or gametes. We get equal information from each parent, and then that information interacts somehow inside this organism. He didn't study the cellular biology here. We have a handle on this already because we're studying meiosis leading up to this. So you can imagine how the traits of one gamete might mix with the traits of another gamete. He's just observing the ratios and the traits of the different generations of peas. Well, how in the world did he accomplish this in the first place? How do you accomplish breeding plants? Right? That takes some care. It's a meticulous work. Breeding animals is fairly straightforward. You put two animals in an enclosure of some type, you leave them alone. It's easy to see that no other animals are in there, so you know who the parents are. Pigeons, goats, you name it, right? Plants are a little bit more difficult. They produce sperm, and that sperm is held within pollen flowering plants, and also some non-flowering plants, angiosperms and gymnosperms, produce pollen. And this pollen is the male reproductive structure that has sperm cells inside of it. And a lot of times it's just blowing in the wind. How do you control and make sure you know who the parent is and who the offspring are? Well, you do that by carefully using a thin paintbrush and dabbing it on the male parts of the plant. So these are called the anthers. The anthers at the top are connected to the entire structure is called the stamen, the male parts, and these anthers on top are covered with pollen. So many flowers have both male and female parts, and pea plants are no exception. We call that a perfect flower. Many flowering plants are that way. A few are not. Some have male-only flowers or female-only flowers, like hazelnuts or ginkgo. But many have both. So here's the male part that produces the pollen, and here's the female part of the flower. And there's an egg cell down here in the bottom. And so he dabs his paintbrush on the pollen, on these stamen, and he then takes that paintbrush and pollinates this white flower's stigma. The top of this part is called the stigma. It's sticky. It's made as a landing pad to receive pollen. And so he takes pollen from a purple flowered pea plant and then applies it to a white flowered pea plant. and then he snips all of the reproductive parts off so that that white flower cannot get self-pollinated by accident, right? These plants will self-pollinate. Many plants will self-pollinate. They can produce pollen, and that can land on their own stigma, and that will cause fertilization and the production of fruit. And we know the end product there are seeds, and then the next generation. So he delicately transferred pollen and then removed those stamens, and then waited for that fertilization event to occur. And as the flower falls off and the pod begins to develop, he labeled these, he collected the seeds from each, and then he planted the seeds. So he crossed these two. Then we would say he crossed them. He crossed these two plants. He bred them together, one with purple flowers, one with white. He crossed them, and that's the parent generation, P. He crossed the parent generation, and that produced the F1 generation, planted from those seeds. So this is time. This is kind of time. It took a lot of time, took a lot of meticulous organization. Okay, and so the results for those were all purple. Well, that's interesting. All purple flowered pea plants. And so if we're to use our terms that we use today, Mendel might have said, well, purple seems to be dominant to white. Purple seems to be dominant to white. Well, that's interesting. And so there's this idea of these traits being in a hierarchy, one dominant to the other. And you get two sets from parents. Well, that kind of fits. We have one chromosome from each parent, and they could pass on the purple version of the gene, the purple allele, or the white allele. Either parent, depending on the parents. In this case, we know that the parent's passing on the purple, and this parent's passing on the white. What does that look like on the chromosomes themselves? This is kind of the language of inheritance here. And we call this the genotype. And so here are our two chromosomes. Here are our homologous pair of chromosomes. And so we have one chromosome from mom, one from dad. And these letters are simply a series of genes. And so we label these genes with letters, or these alleles with letters. And so a different version of a gene is called an allele. And so these two chromosomes, one from mom, one from dad. In this case, if we look at gene A, both parents are passing on the same version of the gene. Both parents are passing on the dominant allele. Big A, big A. Capital letter is dominant. In the case of the next gene, and these could be any traits we're talking about here. It could be all these pea-flowered traits or brown bear versus polar bear traits. In this second one, one parent passes on the dominant allele for B, gene B, and one parent passes on the recessive allele for B, little b. And so there are multiple possibilities. We could get dominant from both parents. You could get dominant from one and recessive from the other. You could get recessive from both. Or you could have it reversed and have recessive from that first parent and dominant from the second. Those two letter sets are called genotypes. And we use those to predict the probability of getting certain offspring. We use that two-letter set that represents the alleles of the organism for any given trait, and we try to use letters that are easy to associate with that trait. So for example, purple versus white flower, we use P. Capital P is dominant for purple, and lowercase p is recessive for white. And we use words to describe those genotypes. And so if those two alleles are the same, we would say that they are homozygous. So they could be homozygous dominant. You got two dominant X alleles, one from each parent, or you got two recessive T alleles, one from each parent. Homozygous dominant, homozygous recessive. Or if you got one from each, one dominant and one recessive, one from each parent, we call that heterozygous. Heterozygous or homozygous genotype. And note the reference there, zygote. We're talking about what the zygote as inherited. The same allele, homozygous, different alleles, heterozygous. Okay, last note today. Here's what this looks like. When we have a set of alleles and their observable traits, you can now see those. We call that the phenotype. It's the observable expression of the genes. The genotype is the two-letter set. You can't see that. You have to infer that after raising the next generation. And we're going to practice that here in the next lesson. The phenotype is the observable expression. And so in the case of flower color in peas, we have three possibilities. We have the purple flower with two dominant alleles, big B, big B. We have the purple flower with one dominant allele and one recessive. Remember, purple's dominant. So if it's present at all, it hides the recessive trait. The only time we ever see the recessive phenotype is when both of these are recessive. Little b, we finally get the white flower. So these are the three possible genotypes and phenotypes for this one trait in pea plants. So that's an introduction to meiosis and Mendel. Stay tuned. Coming up next, we're going to be talking more about how these work and doing some probability using the famous Punnett square. Thanks for listening, and we'll see you next time.